So I have the pleasure of introducing our seminar speaker today, our very own Dr. Jorge Monteiro, who's going to be closing up our invited speaker portion of the seminar this semester. So Jorge did his bachelor's as well as his master's at Sao Paulo State University in Brazil. And from there, he went on to do a PhD at the University of Campinas in Brazil, where he studied similar things that he's going to talk about today, so lanthanide complexes. And from there, he went on to do a postdoc also at the University of Campinas, and then eventually came over to the University of Nevada, Reno. And he has been with us since 2019. He's currently an assistant professor in our department doing research with undergraduate students, which I believe he's going to talk with us about today. So, Jorge, the floor is yours. Take it away.
Thanks for the introduction, Chris. Let me share my PowerPoint with you. Okay, very good. Let me take a page of your book and use the laser here. Okay, so I'm going to talk about a little bit of my research program at HSU. So I'm going to focus on lanthanide luminescent complexes for cellular luminescence imaging and sensing. Okay, so during the talk, to start the talk, I will give you a broad introduction, and as the talk progresses, if needed, we can add a little bit more of the introductory part. Okay. So cellular imaging allows one to diagnose diseases and unravel biological processes. So it's a fundamental thing that contributes to our understand either in diagnosis or on the biological processes. And there are two major ways to image a cell or a tissue. The first one is cell staining. So we are going to rely on a dye and look at color contrast, basically. So doing here an analogy, imagine that the grass is a cell, for example, and we have here a dye that we are going to put in the cell, and that's going to stain some specific part of the cell, okay? So therefore, we rely on color contrast. So this is a very hard technique to get good at. So you need a very good trained eyes to get the color contrast and usually high concentration of the dye. The second way to do it is using the luminescent properties, okay? So now imagine that the dye is luminescent, so we can shut down the lights. So now everything is dark, and you can clearly see here your luminescent dye. So therefore, cell luminescence imaging is much more sensitive than cell staining, and that's going to be the focus of the talk. With cell luminescence imaging, we can track, for example, the concentration of GSH in the cell as a function of the time. We can use for in vivo luminescence imaging and even pH sensing in vivo using emission lifetimes in the NIR range. Okay, so very versatile technique. And we are going to try to connect imaging with a second property. Okay, so what does a compound need to be used in cellular luminescence imaging? It's desirable high emission lifetime, okay, in the microsecond, millisecond, or second range. Of course, we need to be soluble or dispersible in water. and ideally high brightness to give high quality image. So it looks simple, but it is a twofold problem. And the first problem in cell imaging is the instrumental limitation. Okay? So most confocal microscopes, the highest excitation energy available is 405 nanometers, so almost in the visible here. So therefore, your compound needs to absorb around that range. And the second problem that we face in cell imaging is the depth of the penetration of the radiation that you are going to use to excite your compound. Okay, so as we can see here, we have here basically absorbance as a function of the wavelength. So moving close to the UV here, there is high absorption by tissues and cells. And as we move to the red and NIR, there is a huge decrease in the absorption by the tissues. Okay, so ideally, we would like to work in this area here where the absorption is low. So that means the radiation can penetrate more in the tissue. Okay, so those ranges here where there is low absorption, they are going to be called biological windows. Okay, so what's out there? So organic dyes, they are very bright. Usually they have high quantum yield, and we can easily modify the structure to increase solubility or to get the dye to go in a specific part of the cell. So BODIPs, rhodamines, cyanines, those are all commonly used dyes in cell imaging. It's beautiful images that we can get. So even color contrast using two different dyes. However, there are two major problems with this class of compounds. First one is a small Stokes shift. Okay, so if we look the peak of the absorption and the peak of the emission, they are very close together. So that means if your laser is centered here, of course, your laser is broad and your emission window is in this range, there is an overlap here that will interfere with the image. Okay, so that's the first problem. And the biggest problem is photobleaching. So photobleaching means that the emission intensity is bleached as a function of the time, and this can be really fast. So for example, here for cyanide dyes, 10 minutes is enough to completely bleach the compound, and then all of a sudden your image will disappear. So very unstable compounds. Okay, so what can we do to go around that and come up with a better compound? That's where we will focus on my research. So we will go with lanthanide luminescent compounds, especially in the 3+ state. So in the 3+ state, we have unfilled 4f orbitals for the lanthanides. Therefore, we may have electronic transitions within the 4f shell. Because the 4f shell is shielded by 4s and 4p orbitals, there is very low influence of the environment, and we are going to get sort of a line-like emission and excitation spectra that resembles an isolated atom. And because of that shielding, we can modulate the emission wavelength as a function of the metal. So for example, gadolinium has its main transition in the UV, and doesn't matter the solvent or what you have around the gadolinium, this is going to, the energy is not going to change much. Terbium is centered in the green, dysprosium yellow, samarium orange, europium we start moving to red, and then neodymium and ytterbium we will fall in the NIR. Okay, so therefore picking the metal will allow us to pick the emission wavelength, so we can combine several metals to get emission in several wavelengths of the visible or even NIR spectrum. Okay, this sounds very good on paper. However, because we have intra-configurational 4F electronic transitions, they are Laporte forbidden, which means it's very unlikely to happen and reflects in a low molar extinction coefficient. Okay, so around 10 or even an unit. The solution to go around this limitation is to coordinate a chromophore. Okay, so an organic chromophore. Okay, organic ligands, they usually have absorbance around, extinction coefficient around 10 to the third, 10 to the sixth. So they are capable of absorbing a lot of light and ovulating an excited singlet. We have a heavy atom here, so we'll favor intersystem crossing. Then a long-lived triplet is populated, and then from here we have energy transfer to the lanthanide, and then the lanthanide will decay in that specific wavelength that depends on the metal. Okay, so therefore that yields usually photostable compounds. Because those are Laporte forbidden transitions, the emission lifetimes are very long. So we can go to micro to milliseconds range. And there is a large sort of Stokes shift. So now our excitation fall basically in the UV visible, and our emission is far away from the excitation. So no interference at all. And again, the emission peaks, the position of the peaks do not change as a function of the environment. Okay, so very, very consistent. Okay, so when I started working in this area, I came up with this super simple europium complex. Okay, we have here a dipicolinic ligand and an H2 group. And this compound is super soluble in water, very bright. And we tested to tag NG97 cells, which are glioblastoma or brain cancer. So you can see here 3 columns. The first one is just a bright field image. The second one is just the luminescence channel. And then the third one is an overlay between first and second. Looking at just the control, just the cell without treatment with the complex, there is a very faint luminescence in the red. And after incubating the cells with the complex, after 3 hours, you can see that they get very nice and red. So meaning the compound was uptaken by the cell, and now we can tag the cells using luminescence imaging. And that was a very interesting work, actually, because this compound is very specific for some reason for only this cell. Okay, so if we have healthy cells, it does not tag. So it was an accident, not planned, but we got a very specific tagging for cancers, brain cancer cells. The biggest problem with that compound is that the absorption band is basically centered at 300, so far away from 405. So there is a very tiny absorption band here, and that one was the one used to get the images. Okay, so although we were able to get imaging, it was not a good quality imaging. Okay, so therefore that brings us to the goal of the research program: synthesis of lanthanide 3+ complexes with absorption in the biological window that can be applied in cellular luminescence imaging and or sensing. Okay, so to overcome the problem of the absorption band being far from the line, the laser line in the microscope, so we came up with two ligands. Okay, so now we are going to modify the pyridine ring and extend the pi conjugation. Okay, so now we have a very, very, very conjugated system. So this should redshift the absorption band. And the bottom part of the ligand, we have oxygen, nitrogen, and oxygen. So this is a tridentate ligand, very strong, that's going to chelate the lanthanide. And the second ligand is very similar, the pi system, and a little modification on the coordinating units. Now we have 1, 2, 3, 4, 5, 6, 7 coordination points. So that's going to be a heptadentate ligand. So we are going to cage the metal and hopefully make the complex more stable. So this gives you a unique opportunity to be a molecular architect. Okay, so how cool is that? You can plan. The paper accepts everything, right? Plan your synthetic route and go to the lab and see if you are a good planner or not. Okay, so for those ligands, it's quite a long synthetic route. Not that the reactions are complicated, it's just usually overnight reactions, and you end up needing to run a column, so that will take a little bit of time, but we can easily get to the target ligands. Okay, so the first one that I showed here, and then the heptadentate ligands that I showed as an option as well. And when you have the ligands, the next step is the complexation with the metal. So in the first case here, we have one metal for 3 ligands. So net charge is 3-. And then the second case is a 1-to-1 stoichiometry. And the nice thing of the complex synthesis is that you can see right away if you are successful or not. Okay, so just the metal by itself is non-luminescent, and then once you have it with the ligands, it's a very bright and very characteristic red emission from the European. Photoluminescence. Okay, so here on the right side, we have the emission spectra for the European complexes. So again, here, you may change the ligand, but the energy of the transition barely changes, okay? And the excitation or absorption spectrum. So now we have a very broad band, and here we have the line of the laser, okay? So although it's not the peak, it gets the tail here, and that's more than enough. And with these ligands, we can also sensitize the emission in the NIR. Okay, so red region and NIR region, they are all possible. Okay. Quantum yields are around 30%, which is very good. So this kind of compounds, anything above 20%, great. And it's very bright, which is what matters for cell imaging. And again, here lifetimes in the millisecond range. NIR, we have shorter lifetimes because the energy gap is lower. So therefore, in solution, it's very easy to quench the excited state, and that reflects in a decrease of the excited state emission lifetime. And once we had those complexes, someone suggested us to do a 2-photon absorption. And always keep in mind that we are always trying to use the biological window, okay? So 2-photon absorption is a very neat phenomenon. So when you do a 1-photon, a regular absorption, it goes directly from ground state to excited state and then emission. So usually you have high energy here and you get a low energy because of the losses. In a 2-photon absorption process, you are going to split the absorption in 2 parts. So you're going to first get halfway and populate a transient state. So that's a very short-lived state. And then with a pulsed laser, you are going to pump a 2nd photon and populate the excited state. Energy is going to be transferred and then emission again. So now all of a sudden, if you have a 1-photon absorption peak at 350 deep in the UV, the 2-photon absorption peak is going to be half of the energy or double of your wavelength. So now you move all the way to the biological window, for example, 700. So with that, all of a sudden, with the same compound, the excitation now falls in the NIR, deep penetration, low absorption by cells and tissues, and that allows for imaging of deeper tissues. Biggest problem here that requires high power excitation sources, and we need expensive femtosecond pulsed lasers to get this process going. So we tested the complexes, and now notice here that my excitation is in the NIR, and we still get emission in the red, okay? So now I'm using low energy, and as a return, I have high energy. So that's sort of anti-Stokes kind of emission. Okay, so this is very neat. It's a very, very, very cool process. To make sure we have a 2-photon process or a 2-step process, what we do is to collect the emission spectra changing the power, and we plot log of the intensity as a function of log of the power of the laser, and the slope will give us the number of photons needed to achieve the excited state. Okay, so all the cases we have nearly a 2-unit slope that confirms the 2-photon process, and it's not an artifact or anything else. When we have a 1-photon process, what we use to compare chromophores is the molar extinction coefficient. When we have a 2-photon process, we are going to basically use the same idea using the 2-photon absorption cross section of the compound. Okay, for the this compound over here, we got a cross section of 857, and the highest one previously reported in the literature was 775. So we beat the record, and that's still today the highest cross-section, 2-photon cross-section for an European complex in solution. This second compound has a slightly lower cross-section, and again, you can use the same idea of molar absorptivity. So here, if you think about, we have 3 ligands to absorb the 2 photons. Okay, so you have 3 times higher probability than in this case with just one possibility here, and that's why your cross section decreases. Okay, so this is per mole. Okay, always per mole. Okay, so that was very interesting, very exciting. So we can plan the ligand to get a property that we need. And using the two-photon process, we can shift the excitation towards the biological window. That's very nice, but what else can we do with the compounds, right? So can we do something else with the same compound? So that it was when we started thinking about viscosity sensing. And viscosity is actually a very important property, and abnormalities in blood or plasma viscosity are indicative of diabetes and hypertension. Okay, so to measure the viscosity in microscopic systems, we cannot use any physical method, so we need some sort of molecular probe. And what we noticed here is that this nitrogen-carbon bond can rotate, okay? So that rotation will either align the rings or have them be angled. And when that happens, what you have is something called twisted intramolecular charge transfer. And there are two physical parameters that change how easy it is to rotate. One of them is temperature, of course. Higher temperature, higher the probability of rotation, and viscosity, okay? So a high viscosity solution will slow down the rotation and vice versa. So the simplest way to confirm that we have here this kind of charge transfer state is to collect the emission spectra at room temperature and low temperature. Room temperature, you have this very broad emission. So that's a very characteristic charge transfer emission. And when we cool down, you can kind of see a vibrational profile here in the emission, and that's going to be a characteristic of emission from the triplet state and not from a charge transfer state. And then the next question is, okay, is it capable of probe or sense viscosity? So then you need to test that. So here, the range of viscosities are not that broad, but we can see here that there is a response of the emission as a function of the viscosity, okay? So that response we can get in the red with the European complex, as well in the NIR with the ytterbium complex. Okay, so this is within the biological window. And we can also do probe the viscosity using that two-photon absorption. Okay, so now my excitation falls in 800 nanometers, and I still have the viscosity response here. Comparing those probes with whatever is in the literature. For lanthanide complex, we don't have much. Honestly, just one other report, and then here just as a sake of comparison and put DPDI. Okay, so excitation here, 1 photon at 400 or 2 photons at 800. The range is between 0 and 200, and we got here all the way up to a ratio of 5.5 fold for the 2 photon. And that was actually the first example of viscosity sensing using a lanthanide complex and 2 photon excitation. Okay. Biggest problem of these viscosity probes, especially organic dyes, is oxygen influence. So we tested the probes in presence of diluted oxygen in the solution and absence. So there is no influence at all, okay? So that's a true viscosity probe where the response is going to be a function exclusively of the viscosity and not a mixture of viscosity and oxygen, okay? So therefore, with one compound, you can use it for cell imaging, and in this case also for viscosity sensing because of the twisted intramolecular charge transfer states. Okay, so now that we have a little bit of time to spare, I would like to go in another direction here and talk a little bit about Teranostics, okay? So how cool would be if we can combine in the same compound the imaging capability and therapy? So that would give a unique opportunity to tell the patient that, okay, you have this disease because we are tagging some sort of cell, but at the same time, you don't have to worry because we are going to treat right with the same compound. So therefore, Teranostics comes from therapy and diagnostics. Okay? So most of the compounds used for Teranostics are nanoparticles where you can decorate the surface with some sort of therapeutic agents. Nanoparticles are very versatile, but they have lots of problems regarding synthesis, and they are usually quickly recognized by the body. So it's not the best platform for a theranostic. When we go to a molecular system, this is much easier to work with. It's challenging for the synthetic part, but we can easily achieve photodynamic therapy with a molecular system if we do a careful design. And photodynamic therapy is a very cool kind of therapy. It's based on the generation of reactive oxygen species, ROS. And one example here is singlet oxygen. The nice thing about photodynamic therapy is that we activate using light. So therefore, cells are unlikely to develop resistance against singlet oxygen. And how is that generated? So again, here we have excitation of the chromophore intersystem crossing to populate a long-lived triplet state. So now we have the same multiplicity for the excited state and ground state of oxygen. So we can do a cross relaxation here and generate the singlet oxygen. So singlet oxygen is very reactive and it will attack anything that's around it. However, it's short-lived, so it will not propagate. So it's a very localized kind of therapy. And singlet oxygen can decay back to the ground state, and the emission is going to fall in the NIR. Okay? So therefore, our goal is to get a compound that's capable of doing the diagnostics and generate a singlet oxygen. So this is very recent. What was the idea here? Again, always looking at the biological window. We know that this carbazole moiety here is a very strong 2-photon absorption, has a very strong 2-photon absorption. However, it cannot generate singlet oxygen. And thiophene, they can easily generate singlet oxygen in high yields, but they don't have strong 2-photon absorption. So you have two pieces of the puzzle. So the simple solution here is to combine both. So these positions of the carbazole, they are very reactive. So it's not that hard to functionalize. So what we will do here is to keep our 2-photon absorption core and add singlet oxygen functionalities. So the hypothesis here is that this here can undergo 2-photon and do an energy transfer to the thiophene rings, and from here generate a singlet oxygen. So therefore, still luminescence, 2-photon absorption, and singlet oxygen generation capabilities using excitation in the biological window. Okay, so this is quite a complex system to synthesize. Again, you plan the synthesis and see what happens. As as you start to functionalize things, you start adding more steps. It's not hard again. It just takes time and trial and error basically, but we can go all the way to the end and get to the complex. And the way we are going to probe or prove the singlet oxygen generation is using a probe. Okay, so we have here this compound in the presence of singlet oxygen. It will attack this furan ring, open the ring. So this is luminescent, this is not luminescent. Okay, and the very characteristic band here is at 480 nanometers. Okay, so therefore, if we can generate singlet oxygen, this will decrease the emission of this species because we are producing a non-luminescent species. Okay. Exciting the sample at 720, what you can see here is a decrease on the emission of this compound. So that proves that, yes, we can generate singlet oxygen using 2-photon and allow here an intramolecular energy transfer and get this going. Okay, so it stops at 10 minutes because you completely saturated the oxygen in the solvent. Okay, so there is no more oxygen to generate singlet oxygen, and that's why you plateau here. And this is actually a very efficient system. It's more efficient than some of other dyes that can do that. Okay, so again, knowing what you are looking for and having knowledge of other areas of chemistry, you can plan things, right? Be a molecular architect. And chemistry gives you the unique opportunity to go to the lab and prove your hypothesis. And in this case, we were successful. So we achieved one of the goals. And I think that's a good point to stop. So to summarize here, I showed you the capabilities of lanthanide luminescent complexes. They can go 2-photon absorption, okay, so sort of an anti-Stokes transition. We can use this particular complex for viscosity sensing using 1-photon and 2-photon, and also in DNIR, insensitive to oxygen, and attaching an extra functionality to the carbazole ring, we can generate singlet oxygen using 2-photon absorption. Okay, so I have to acknowledge a lot of people here. I'm the one proposing things, but who is doing the work are the students. Okay, so Jessica has been for a long time in the lab. So lots of things here helped me set up the lab. So we have Gage. Gage does a project that Claire and I overlap. So he has the opportunity to work with 2 different advisors in 2 different areas. And Ben started last semester. Elizabeth worked for 2 semesters, but sadly she graduated and left the lab. And thanks for all the collaborators and for all the sponsors that believed in the ideas and supported the ideas. So with that, I will end the talk.
Okay, thanks to George. That was a very interesting talk. Very cool. I'm sure George would be delighted to entertain any questions. We've got plenty of time. So I'll ask the first question since everyone's being shy. So the two photon laser you described, so how do you have to do a lot of work with planning the rep rate, the repetition, the pulse rate, compared to the states of the system, or you just give it a huge blast of photons and hope that it works?
Yeah, you just give it a blast.
Okay.
And kill a chicken, right?
Yeah. And so that's why the intensity was so much less than the one photon?
Oh, definitely, definitely. It's a very low probability setup because you have that transient state. Mm-hmm. And it's very unlikely to first exist and second to populate and populate again to go over.
I had a question about the same laser, which was, why does it have to be femtosecond pulses to make the double?
Because let me go back here. That's a very good question, Claire. Allow me to share this again. Let me go back here. Okay, so what you have here is a sort of electronic state that kind of does not exist. So you have to pump once, and once this is populated, you have to pump it again. So if it is a continuous laser, you are going to pump here and decay back. That's the first problem. The second one is that when you pulse the laser, what you are doing is to accumulating a lot of energy and boom, releasing in one shot. So that gives you a huge power density, and the laser is going to be focused on the sample.
Okay. So like then you have more power coming right at the moment when you have that transient state. Is that true?
Exactly. Cool. And Chris can correct me if I'm not wrong, if I'm not right.
What I was going to ask was actually, is that kind of like a Rayleigh scattering event? This
it's
virtual...
a Raman, right? It's a Raman.
A Raman, yeah.
Yeah, it's an anti-Stokes Raman. Yes, exactly.
So it's really like, it's not like an actual state, but it's really like the electronic environment of the molecule that it's scattering off of.
Exactly. You combine these two wave functions, right?
Yeah.
Using the polarizability operator, and then you produce this midway state.
Yeah. And you have no shortage of electrons floating around in those compounds to...
Exactly. That's why you need to get the conjugated system like very, 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 very conjugated.
Yeah.
I have a question, George. In your human skin, so you have this analytical window that you are dealing with, that you want your emission peak to come in. What are the existing peaks in that window? You have two peaks there. What do those correspond to?
Let me go back here. Share this again. So this one and this one?
Yeah.
Okay, so this is water absorption. And if I'm not wrong, this is water as well. I'm not that sure, but I'm almost right that those are the two water peaks.
Okay. And then my other question was, so you excite your complex at say 300 or so, and then you get emission at 400 to 500 in the visible range, but then you get another set of peaks in the infrared. So yeah, I've never seen phosphorescence described like that. Why do you get the second set? What is going on with the energy states, and why do you get that second set that's shifted to the infrared?
Let's see here if we are in the same page. Like for example, this here, Matt. Yeah,
and then you get at 700 you get a little extra, or or even the pink over purple over there, you get another set.
Yes, exactly. So this is for European. Okay, so all those narrow transitions, they are FF transitions within the European. Okay. And this one, we change the metal. And now what I'm using for the NIR is ytterbium. Okay, so see here. So because we have different amount of F electrons, you are going to split everything in a different way. So we fall in the NIR. And for European, the main transition is here, but we can go here, 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 and here. But it's mostly in the red, and this is mostly in the NIR.
Okay.
George, is it oversimplified to think of that as like, the classic particle in the box, like what happens with quantum dots, like the bigger the system gets, the more red shifted it becomes?
Exactly that, Chris. Exactly. So you can imagine, like if you don't have the carbazole, the length is going to be all the way here. When you add the carbazole, you increase the length of the box, and that decreases the energy of the transition.
So what kind of laser system do you have here? I don't think that has a femtosecond.
No, with the one we have here, cannot do it. And the 2 photon is very nice, but it's super expensive.
Yeah.
Femtosecond laser is going to cost 100K, 150, and it breaks all the time. Very expensive. So I would rather go to a collaborator that deals with that and just give the complexes.
Very cool. We've still got plenty of time for questions. Any questions from the students?
I'll ask one. So George, why does the glioblastoma pick up the compound and not a normal cell? Do they know, is it an efflux pump issue, or do they know what it binds to?
I have no idea. And we collaborated with another group to do that. And they were not interested in mechanism or the reason why.
Seems like it would be something interesting to do a 2D gel on and then see what in the gel picks up.
Yeah, I don't know if it's the charge or if because you have the NH2 group here, you bind to something and that facilitates to get uptaken. I guess
you could tell if it was an efflux pump type of thing that went away if you lyse the cells and it doesn't work anymore.
I see. I mean, I have enough of the compound here. If you wanna,
I don't. It would have to be somebody growing cells, probably though.
Yeah.
I'll grow bacteria, but.
George is happy to dish out his little vials to anybody that wants to use them, right?
Definitely.
Okay, any other questions for our speaker? Okay, very good. Let's give George a virtual round of applause. So just I have a